Welcome, everybody, to the California Tidal Marsh Restoration Science Symposium, where we are sharing lessons learned from Hester Marsh in Elkhorn, Slough. I'm now going to introduce my boss, John Battistoni, who is Elkhorn Slough's Reserve Manager, to say a few words.
Hi, good morning. As the slide says, and Kerstin just said, I'm John Battistoni. I'm the new Reserve Manager here at Elkhorn Slough. I'm now halfway through my 6th week as a Reserve Manager, so I'm very happy to be here to be able to listen and learn along with everybody else here about all the hard work and the great science that's gone into the Hester Marsh project. I just want to personally thank everybody who's putting on the symposium, the ESNR staff, all of our speakers today, and especially want to thank all of those who have contributed to the project and making it what it is and where we're at now to be able to talk about it today. So thank you everybody again for coming, and want to welcome you all virtually to Elkhorn Slough.
Thanks so much, John. Yeah. So today we're going to be focused on the Elkhorn Slough estuary located in Central California at the midpoint of the Monterey Bay, specifically here on this area, which is Hester Marsh before restoration. We'll be talking about restoration efforts to undo harms caused by human activities over the last 2 centuries. You can see what some of those might be from this aerial image. We want to recognize that prior to colonization, the estuary and the lands around it were stewarded sustainably for millennia by multiple tribes that called this area home. Colonization resulted in devastating consequences to the estuary and to the people who had lived here since time immemorial. We at the Elkhorn Slough Reserve and Foundation are partnering with multiple tribes in the area today and support their efforts to reconnect with these ancestral homelands, and we really honor their legacy of wise coastal stewardship. So today, our objective is to share what's been learned about restoration approaches, successes, and challenges here at Hester Marsh at the Elkhorn Slough National Estuarine Research Reserve. We have 2 types of target audiences and outcomes for them in mind, and I think that's really well represented by the participant list I see here. One is coastal managers and scientists from throughout the country who are interested in applying the lessons we've learned to other estuaries. The other is our own local Elkhorn Slough community, so they can understand what has happened in their own estuary. We have various different sessions today, sort of color-coded here, this overview first, then some broader habitat talks, some experimental restoration talks, and a blue carbon session after lunch. We'll go straight from the first to the second, but then there will be a short break between the other ones. So it's a packed schedule full of great talks today. And our first speaker will be Monique Fountain. She's worked at the Elkhorn Slough Reserve for the past 16 years, serving as the director of the Tidal Wetland Program for the past 13. She brought a wealth of experience to the job, from her background in marine invasions and genetics to being a certified project management professional. Remarkably, she's helped to bring in over $26 million to support restoration and science on the Elkhorn Slough Reserve. And she's served as the project director on the Hester Marsh Restoration Project that is the focus for today. I can truly say that this restoration project would not have happened without her leadership. She had the unique skill set needed to navigate the collaboration, the design, the permitting, and implementation of this major restoration project. Moreover, she had the courage, the patience, and the flexibility to overcome the inevitable challenges that arose. She really has a knack for improvising creative solutions to problems. So it is an honor to introduce my colleague, Monique Fountain.
Thank you, Kerstin. And thank you all for coming. I'm a little bit speechless from the introduction. I appreciate it. I will be giving the overview of Hester Marsh project today. And let's see, and so I may be giving this overview, but as John Battistoni said, everyone at the Reserve has had a hand in its success one way or another. And I just feel incredibly lucky to be part of such an amazing team doing all of this great work. Quick reminder where Elkhorn Slough is. It's about 50 miles south of San Francisco Bay in the middle of Monterey Bay. 7-mile long coastal embayment emptying into a branch of the Monterey Bay Canyon. And the Elkhorn Slough National Estuarine Research Reserve is one of 30 sites throughout the nation designated to protect and study estuarine systems. They were established through the Coastal Zone Management Act. Each reserve represents a partnership between NOAA and a state agency in that state. At Elkhorn Slough, this is the California Department of Fish and Wildlife that owns and manages the reserve. And you met John Battistoni, our manager. The third partner at the Elkhorn Slough Reserve is the Elkhorn Slough Foundation. The foundation is an accredited land trust conserving over 4,000 acres around the reserve and serving as the fiscal agent for the NER funding from NOAA. And as part of the NER system, we focus on place-based science with long-term monitoring used to inform management. So like other estuaries along the coast of California, Elkhorn Slough is a rich ecosystem with a diverse array of species and complex food webs. It's salicornia or pickleweed dominated with no spartina or cordgrass. Portions of Elkhorn Slough are designated a Ramsar site, marine protected area, state ecological reserve, and a wildlife management area. The slough is also an important stop on the Pacific Flyway for migratory birds. This unique environment draws a lot of visitors to the slough, creating an economic engine driven by ecotourism. However, it's very much, so this makes this a very high value system, both ecologically and economically. However, it's very much set in a working landscape with extensive historical modifications, as Kerstin said, such as the diking and draining of wetlands, as well as future threats from climate change, especially sea level rise. So as a NER, the long-term data sets and the short-term studies have been vital in characterizing and understanding the changes we've seen, as well as guiding our approaches to future restoration. So this includes understanding what was here in the past, the historical ecology work done by Andrea Woolfolk, characterizing the physical changes such as subsidence and cumulative impacts of sea level rise. Here's Charlie Endrest doing a surface elevation table. As well as interpreting the biological interactions, such as how otters use tidal marshes. So I bring this up to let you know that today's talks are built on a strong foundation of over 40 years of dedicated science in this estuary. And the work you'll hear today serves as additional building blocks in our understanding of this system. So let's look back in time for some context on the system, and then I'll cover our process of doing a project at this scale. So this reconstruction from 1870 shows the extent of our wetlands at that time. Blue is water, brown is mud, and green is salt marsh. By 2000, Elkhorn Slough had lost 50% of its tidal marsh, and this was largely due to the diking and draining of wetlands. So this legacy of diking persists in a system with low sediment supply and subsidence. So this slide shows a small portion of marsh that was never diked up in the top, and the rest is a historically diked marsh that was subsided. Even after tidal exchange was returned, the formerly diked subsided areas remain mostly too low to support marsh. Marshes are not built up over time by sediment since the river was diverted from the estuary and they're actually subsiding. So you just have these fringing edge of marsh around the outside. So we lost a lot in the past, and unfortunately for the marshes that remain, things are looking challenging for the future. Many of our remaining marshes that weren't diked are not currently tracking sea level rise. They're gradually sinking, and modeling suggests it won't take much of an acceleration of sea level rise to drown them. So here's the current distribution of various habitats in Elkhorn Slough. Focus on the green, that's the right elevation for marsh. You can see where it is, mostly in these areas that were never diked. This is a shaded area, are areas that either are or were formerly diked. So this is what 50 centimeters of sea level rise would look like. Almost all the green is gone. Not much migration because the slopes of this estuary are relatively steep. So this is a critical topic for decision makers. How can we ensure that key habitats and their ecosystem services are preserved into the future? So in other words, what's the process? How do we get it done? So here at ESNR, we have the Tidal Wetland Program. It's a broad group of agency representatives, scientists, and community stakeholders that drive the trajectory of restoration through a collaborative process that started in 2004. So many of you have been integral to this process. And we looked closely at a number of different options, including conserving existing tidal marshes. So where possible, we try to protect our marshes from stressors like eutrophication and algal mats. But again, the modeling suggests that we'll not be able to protect our marshes beyond about 40 centimeters of sea level rise. So facilitating marsh migration to higher ground, for the most part, this is not an option for us because we have relatively steep hills next to the marsh. And thin layer sediment addition. This works well if you have a regular source of sediment from a nearby channel dredging. We don't have continuous channel dredging going on here. It just happens every 5 years or so. So it's probably not a good option for Elkhorn Slough. So the thick soil addition, however, proved to be the winning solution from our strategic planning. And by adding a lot of soil, we could restore elevation capital that had been lost due to diking and draining, and at the same time, build the marsh high up to be climate ready. So most of our soil was scraped from adjacent fallow farm that we'd acquired. And so this gave us a chance also to recontour those steep hillsides into some gentle slopes that allow for migration. So going back to our slough overview, we chose a place called Hester Marsh. So here, formerly diked and drained. It was a former farm also and had easy access. And yeah, this is an area that had been diked and drained. So looking at the historical changes over time, here's 1931. It shows the marsh that was there. And then in 2012, it shows the changes of just, again, the little fringing marsh around the outside. And so we have a bit of a blueprint to work from, not really to restore to any point in time, but to restore those ecosystem services and bump the system back into balance. So we relied heavily on a number of working groups to provide guidance during the design of the project. And for example, we had a hydrology working group that evaluated tidal channel morphology, and the salt marsh working group that helped us set the elevation and the channel density and the slope of the marsh. And I've had a drought working group that worked through the challenges of restoring a wetland during drought conditions. So thank you again for all of you that participated in a working group. Just know that your input was really critical to the success of the project. And I think I'm just going to do a quick segue here to permitting. I think the science-based open collaborative process really helped us navigate the permitting process. So don't really look at this slide. This just is kind of to illustrate how crazy it can be. I think everybody here knows that it's a very long and complicated process to obtain permits for this type of work. It took us the better part of 3 years to obtain all the state and federal permits. And I think involving all of these agencies in the planning process was really key to getting this done. And one person that guided us through this, April Zahn from Ducks Unlimited. I hope she's on today. Very helpful, and really, this is a chart that she made, and it's just super helpful to follow this incredibly complex process. So back to the project. Our working groups helped us put together and define our restoration goals of restoring, this is 61 acres. We actually did 122 acres of tidal marsh. This was just for the first phase. Protecting and improving water quality, reducing tidal scour in Elkhorn Slough. So tidal scour is that scour of water moving in and out with the tides. If you fill an area, then you're reducing the volume of water in the slough by that much fill, so you reduce the tidal scour. Increasing our understanding of blue carbon, which you will hear all about from Dina Payton's group later in the afternoon. Improving southern sea otter habitat. So we have the largest concentration of mothers and pups, southern sea otter, in this area here. And then we restored this another 61 acres here 30 acres here and 30 acres over here. So this area adjacent to where they are and hoping they move in there. And then improving resilience to climate change, which is really important. So what we're talking about here primarily is sea level rise, because it's all about elevation. So here are the zones. We have our tidal creeks down here, low salt marsh, high salt marsh in this darker green, marsh upland ecotone, and coastal grasslands. So one of the reasons that we understand these this relationship so closely, and we can actually put all these numbers here, is because of the NERSC Sentinel Site Network, which was designed exactly for this, to gather precise data about changes in elevation and inundation to inform research managers about the effects of sea level rise. So in Elkhorn Slough, we know what our marshes look like when they're too low. This is very patchy and not particularly healthy looking plants. And we know what they look like when they're too high. They're fields of weeds. And we really wanted to set the marsh as high as possible in the tidal frame, right up here, so that we could give it as much of a chance to survive through sea level rise changes. So in terms of setting the elevation, they say that every great idea started on a napkin, and here's ours. Well, it's actually Ivano Ayala's from Wasson Marine Labs. It's apparently not enough to just throw some soil out there to bring the elevation up, because there will be consolidation of the soils you place on top and compaction of the old mud below. So this is Ivano's mock-up of the marsh soil profile, and understanding this profile gave us a target elevation that, when it settled over time, it settled to our final desired elevation. And so we had to overfill the marsh to get to the point that we wanted to, but we couldn't overfill it too much or we'd end up with a field of weeds. So I have a quick little video, actually this, just to show, let me go back a second here. This at the beginning. So this shows what it looked like before the restoration. Here's Yampa Marsh. It is a drowning, a fairly low marsh, but you can see how much more lush it is. So then just some construction photos to show you. So it was moved a truckload at a time to get out there. So it was a fairly long process. Each phase of construction was about 9 months long, and really want to take the time at this point to call out all the contractors we worked with. H.T. Harvey, ESA, and Moffitt Nichols helped with the early designs, and then Ducks Unlimited has really been an amazing partner throughout the rest of the design and as our construction administrator. So Steve Carroll has been our engineer and has really been here through this entire process. It's just been amazing. So here you see they filled the whole area. They used about almost 500,000 cubic yards to fill it, and then when they opened it up, we were super excited to see right after it opened up that we got wildlife coming in, all sorts of different kinds of wildlife. So very exciting for that first part. So then in borrowing from the hillside for the marsh restoration, we borrowed material, but we also had some material from the Pajaro River. To the north is a flood control project, and we got some of the material from there. But in borrowing the soil from the hillside adjacent to the marsh, we were left with 35 acres of scraped hillside, and our stewardship team, led by Andrea Woolfolk and Mary Paul, have been transforming this area back into a native-dominated coastal grassland. So they collected seeds and rise zones, and the team experimented with a number of different planting methods. So in general, they found that hand seeding was more successful than broadcast seeding with a tractor, which was interesting, and that plants that grow by rhizomes, like creeping wild rye, generally do better over the long run. So that's been a really exciting process. And here's the borrow area, and the area that's this bright green area is where the stewardship team is working hard to bring back those native grasses. So in total, this area is about 120 acres of marsh and 35 acres of grassland. And the project was done in 3 phases, with each phase monitored really intensively, and those lessons learned applied to subsequent phases. So phase 1 was open to tidal flow in August of 2018, phase 2 in December of 2021, and phase 3 just last November 2024. So these images are digital elevation models of how the marsh was transformed from a formerly diked and degraded site to a very high marsh climate ready. So these blue, these are the tidal creeks and sort of mudflat elevation. This green is low marsh, and then the darker green here is really high marsh. And this is before, and then after the restoration, you can see the whole area was high marsh. We brought it way up there in the tidal frame. And we also added about 14 acres of marsh along this edge here. Okay, so you'll hear more about this later, but colonization by vegetation takes time. So when building such a high marsh that's designed to persist in the face of sea level rise, you create a relatively harsh environment, and it takes time for vegetation to colonize in this harsher environment. So Kerstin and the rest of our speakers will cover various experiments looking at the mechanisms in more detail, but I wanted to cover some of the large-scale adaptive measures we took to try and mitigate these harsher conditions. So here in particular, you can see that the channels, which are lower in elevation, have re-vegetated relatively quickly, but it's patchy on the top of the marsh plain. And we realized that the heavy equipment, which did such a great job of compacting everything and allowing the contractors to dial in that elevation so carefully, may not have been great for the plant recruitment. So one approach was to decompact the soil. And this helped a lot in phase 2 as pickleweed seeds settled and then sprouted in these furrows. And you'll hear more about that. If I can get to our next slide. Let's see here. Okay, so another approach included salvaging pickleweed that was going to get buried. So these root balls were moved with tractors, this tractor size moving of material to see if the plants would take. So this is just, they weren't laid as a nice piece of sod. They were just put in there at the correct elevation. It was a mass of roots. And then, a couple of years later, they just came out really nice. And you can see how much nicer this is than the surrounding area. So transporting these plants wholesale, really, really great. And then retrospectively, we noticed that areas with more organic topsoil had better recruitment. So we incorporated this into both large and small experiments. And so phase 3 has a lot of large and small experiments built in. And in the time I have left, I'll take you on a quick virtual tour of this phase. It was just open to the tides last November, and we'll visit a lot of these different little areas here. I'm going to go along this way and then back along the edge, back in here, and then walk out through here to look at all of these different little pieces. So let's get started. We're going to run through this virtual tour. All right, so one of the features that was really neat here was this raised channel edge, and this was to reduce erosion on the outer edge. And this was raised up just a few inches, ensuring that when the tides came in, they would fill in from the internal channels and go out through the internal channels. And sometimes it did go over the top, those really high tides, but in general, the idea was that it would go in and out through those channels. And then we're going over to this first crop. So we wanted to try a big experiment of using that organic topsoil. So this plan was we put in 2 feet of topsoil in this area. And originally we wanted to put a nitrogen fixing cover crop in here to grow those plants, fix some nitrogen in the soils, and then put some pickleweed seeds in. And because this area was diked and we got a tremendous amount of rainfall that winter, the whole area filled up and was a lake had about 6 inches of water in it for that first winter. And that destroyed the cover crop because there was still salt water in some of those tidal channels. And then some of the pickleweed has come up. So you can see that. And then kind of moving along, we have an area where you can really see where it was decompacted, soil decompacted there. And some of these really nice looking channels that they did. I'm kind of walking through here. It's kind of fun to do a fast field trip. Okay, so this area we're coming up on here is an area where marsh was salvaged, and it was placed in this area with a dozer, and then it was spread with a dozer because that really is probably the most cost-effective way to get it out there. And then they did decompact this section here But of course, that meant they were driving over it again. And so we had another section over here. We asked them not to rip or decompact. And you can see the difference there. And then moving along out here, we have a surface elevation table that Charlie put in. We'll be monitoring changes in the elevation of the marsh with that and with the drone imagery that Charlie takes too. All right, so you can see some of these nice looking individual plants coming up. They're doing pretty well. They look pretty good. Now we'll go out towards the edge of the marsh here. Of course, had to stop and talk to the kayakers because they have questions. I think most of our outreach really is to kayakers when they see us out there, and as well as the groups that come out. And so this is kind of interesting, the slurry area, and I wanted to point out these runnels that have already formed here to kind of drain this area. So this was a slurry that was put in out of a tidal channel because in the hopes of rich in organics, hopefully had a seed bank. We'd seen in past phases that areas that had this slurry, these cracks trapped the seeds really well, and they grew, and they were some of the first marsh plants to show up on the marsh plain. And apparently that didn't happen here. So we're watching this to see what happens. Then we also have an area where we salvaged marsh we've spread with a dozer, not decompacted, but it's looking pretty good. All right, we'll head out to the edge, look at the firm channel edge. This is a feature that Steve Carroll came up with to protect against erosion. This firm channel edge has packed bay mud along the edge here. This is really where our prevailing water comes in and where most of our erosion will occur. And this area's been open for about 10 months now, and you can see it's doing pretty well. And then we're transitioning into another area where we just have more of a slope, so 10-to-1 slope on the bank here. It's just of soil. It doesn't have bay mud in it, and this is also doing really well. Nice little slope to reduce that erosion effects. And we have our sign out here because this really is, like I said, where a lot of our public outreach happens. And so, again, to thank our funders, California Department of Fish and Wildlife Greenhouse Gas Reduction Program, California Department of Water Resources Integrated Regional Water Management Program, California Ocean Protection Council, California State Coastal Conservancy, California Wildlife Conservation Board, the NOAA Bill Habitat Restoration, U.S. Fish and Wildlife National Coastal Wetlands Conservation. And so in the interest of time, I'm going to go ahead and jump over the eucalyptus trees over here. And you'll see this area then was salvaged marsh that was spread with an excavator. So this is a very careful and deliberate process where it's been moved over and placed in here. It looks good, but it's very, very expensive to do. It just takes, because you're handling the material for a long time. So before we head out further, there's a little area off here to the left I wanted to look at, which is where we tried to do, this was my second attempt at a cover crop. So this time I diked up a little area and I had the cover crop planted. And of course, the first time it overtopped, it actually soaked through this dike, got this area salty. So the cover crop again didn't make it. I'm open to ideas. If you have any ideas how we can do this cover crop successfully. But what I did at the end was I just threw all of the seeds that we had, and you'll hear later from Bethany about working with pickleweed seeds, because one of the dreams is to collect a bunch of seeds and go out and sow the seeds and have a marsh come up and then open it to the tides. And so this is the first time we've got some seeds coming up. It's super exciting to see all of these little tiny seedlings coming up. So hopefully we'll have more success with that in the future. And then heading out along the edge of the eucalyptus grove here. See if I can turn around, and there we go. And this area is really interesting. So you'll notice that this area really is looking pretty good. It's more lush. We have a lot more plants that recruited in here. And there certainly is water coming off of the hillside. So you'll hear also a little bit about the importance of water in our marshes. So this is more salvage pickleweed that was spread with an excavator, as well as these two plots here. Tried a couple of different things with doing that, and you can see how really lush they look, but again, super expensive. Here's a look at our little rookery. We have herons, egrets, and cormorants nesting here, and they were all talking to me as I was walking through. It's kind of fun. And then this last little area here is marsh that was preserved in place. So it happened to be high enough that they could just work around it. They put a nice tidal channel in here so it could drain. And it, of course, looks fantastic and will serve to help propagate the rest of the marsh here. So that is a really quick tour of phase 3. And also, oops, see if I can get through to the end of my talk. So any questions? I'm going to stop sharing so I can see the chat. Kerstin, can you help me with questions?
Yes, I have been helping. People
Great.
asked about how we chose our creek densities and locations, which I answered that we looked at existing creek densities in Elkhorn at Good Marshes and the 1931 aerials. Where possible, we mimicked those. And then there was a question about whether furrows might have sort of persistent hydrological effects that might be undesirable over time. And I'd love your perspective on that. My thought was they seem to be flattening pretty fast, so I think it's going to be short-lived, but I don't know.
They do, and then the runnels that you saw there will seem to be emptying them out. And so we put them in a way, I don't know if you noticed, they were sort of, again, they were parallel to the tidal creeks. So they weren't emptying directly in, which helped keep the soil in, but then those runnels broke through. The water will always find its way and create a path. So I think they do flatten pretty quickly.
Lots of compliments on the virtual tour, which I know took some time to put together, but sounds like it was worth it.
It was an Insta360 camera that John Haskins got, and yeah, it's a little steep learning curve, but it was really fun.
There is a question about, after Hester, what's next for the tidal wetland program?
Oh, well, yeah, thank you for that question. So after this, we'll be looking at Pelican Marsh, which is up near Kirby Park, and that was a really challenging area because it's sandwiched in between a railroad and a road. It was a formerly diked wetland, so it'll include looking at infrastructure, and yeah, it's exciting. We have some funding to start working on that through the California Marine Sanctuary Foundation grant. That's a regional challenge grant from NOAA. So we're really excited to get started on that.
Question on the Parsons sill, and you might need to explain what it is for most people who won't know. How is the sill doing? And
the sill is still there. So the sill is a 280-foot long sheet pile wall that is integrated into the railroad at the mouth of Parsons Slough, and it was designed to reduce tidal scour in the estuary, and it has reduced the tidal scour downstream of the estuary. And yeah, I think that we continue to monitor it, and we gave an update. We do have an update on it's been a while though. It's been a few years since we've done it, so maybe we have to take a look at that again and give you guys an update. Thank you for bringing that up.
Question on soil monitoring for peat formation. She'll be kind of answered this afternoon, but you could give a foreshadowing.
I think, yeah, go ahead on that one if you like.
Yeah, I mean, so peat is laid down in centuries of marsh growth and tracking sea level rise. When you take cores in our existing marshes, meters down, we have peat equivalent to thousands of years of marsh history. Hester is brand new, so the root zone is only 10, 20 centimeters deep, and the plants are small, and the vegetation cover is light. So yes, we are starting to put down that future, tomorrow's peat, but we are at early stages of that. Any last questions? We have time for one more question if somebody has one. Are you experiencing any issues with weeds in the high marsh?
We are. We did. There was a student of Kerstin's that looked at exactly that zone where that zone of the ecotone and sort of where pickleweed sort of petered out and where the upland weeds came in. And we chose an elevation where we had about 10%. We expected about 10% weed cover, and that's about where we were at, maybe a little bit more at the very top. And we had a lot of problems with weeds, brass buttons and things in phase 3. It came in very strong, rabbit's foot, but then the first few tides, I had noticed that the first 2 tides had wiped those out. So I think it's a trade-off. All right. Looks like we are moving on here. So we're going to move on now to Kerstin and hear about lessons learned. She's going to introduce the rest of the symposium and also talk about lessons learned from collaborative science to restore estuarine foundation species and habitats. And Kerstin is the research coordinator at ESNR for the past 25 years. She's also an adjunct professor at University of California, Santa Cruz. She's published over 60 papers on Elkhorn slough science in peer-reviewed international journals. So in addition to her own science that she's doing, she serves a really important role luring others into the mud at Elkhorn Slough. She is cheerleading and supporting their research, and like many of the other speakers today that you'll hear from. So in 2004, she co-founded the Tidal Wetland Program with Becky Suarez, who I saw was on the call today, recognizing the need for estuary-wide strategic planning. And that's a lot of what Tidal Wetland Program did for the first 6 years that it was in existence. Kerstin serves as the science lead on the Hester Marsh restoration, and she will now share what she's learned from being in that role over the past decade. And I also can say that she is just an amazing mentor and scientist. So it's exciting to hear her updates.
Thank you, Monique. So for the next half hour or so, I am going to be sharing with you in a big picture sort of way what we have learned from restoration science at Hester Marsh. The rest of the symposium will have more detailed research talks on components of that, but I thought it'd be good to just step back and give an overview, a high-level overview, for which I found this photo to illustrate the 30,000-foot view, which was out of the window of my Southwest airline flying back from LA. And I've got to say, it's sort of exciting and sort of terrifying when you can see this marsh we've built out your airline window. I want to start by paying tribute to someone who has been a lifelong inspiration to me, or career-long inspiration, Joy Zedler. She conducted pioneering salt marsh restoration research in Southern California and was really a tireless advocate for incorporating large-scale, well-designed, replicated experiments into all the restoration you do. Restoration's inherently experimental. Why not learn from it in some good experiments that are built in? So I think you'll see the Zedler influence in my talk and in the rest of today's talks. As Monique mentioned, large-scale experiments can be built right into the earth-moving stage with heavy equipment, such as in phase 3, which you just saw the virtual tour of, these different sediment blocks and pickleweeding spreading blocks that we are studying with replicated plantings and UAV. You can also build large-scale experiments into the planting stage. We've planted around 50,000 greenhouse-grown marsh plants into Hester Marsh, and most of them were a replicate in one experiment or another. So you always see in the early stages flags all over Hester Marsh. Experiments can also be added as a part of later adaptive management, trying to maybe decrease salt stress with sprinklers or doing direct transplants of creek plants into bare areas. These will be topics of 2 talks about an hour from now that you'll hear more about. So what have we learned from all these experiments? Again, I don't want to spoil the takeaways of the later talks, but because they're detailed, zooming in on different aspects, I thought it'd be helpful to tell you sort of the most clear messages we've gotten from all the experiments we've done at Hester. Probably the strongest statistical effect we've seen is not a very conceptually thrilling one, which is that large plants do better than small ones in stressful conditions. Rachel Pausch did a study comparing 2 sizes of pickleweed, and the large did a lot better than the small. And you'll be hearing from Susie and Melanie today, who looked at even smaller and even larger sizes of pickleweed. And overall, this rule holds true. Big is better if you're working in stressful soils. We also tested 2 different sizes of 3 other high marsh species and found the same result as you can see in these Frankenia pairings here. Another really clear message was about those furrows that Monique mentioned. Furrows really were great for wild seedling densities and for transplant success. So they were better than the adjacent ridges and better than unripped areas. So that was a really clear lesson takeaway for people working in compacted sediment addition sites, is ripping is good. We were also really interested in intraspecific interactions and the potential for clustering to provide some facilitation benefits in a stressful environment. So in phase 1, we planted 19,000 plants of 7 different species, divided evenly into clustered versus uniform sort of belts, transects that spanned an elevational gradient. And either there were 9 plants planted very close to each other or 9 uniformly spaced. And this was work Karen Tanner recently published. So what did we find with this? As we tracked these over time, you can see that in the clusters, this is later intervals of the same clustering, the clusters bumped into each other and competed, while the uniform plants had more time to spread out, more space to spread out. So from a restoration practitioner perspective, uniform was better because there was a lot less bare space than in the clustered. We really didn't see much benefit to clustering and conclude that competition outweighs facilitation, so managers don't cluster your marsh plants if you're in a system like this. We've also looked at interspecific facilitation and have found that it may provide some benefits. You'll hear from Ari about Grindilia and Emma about Frankenia maybe playing a role in this regard. We did experiments on watering and on soil amendments that Anna will be talking about in a little bit. And we found that they only provided minor, if any, benefits to transplants and wild seedlings. And let me just pause there. Are you surprised by that? We were surprised. When you have a really high marsh that's hardly ever inundated, you would think that running sprinklers, you'd get like a green box in here of really happy vegetation. Likewise, in a system that has maybe 2% organic matter in this soil we added, you would think adding organic soil would make the plants really happy, but wasn't really what we found. So another lesson I want to share with you is that the results of your experiments may surprise you, which is exactly why you need them. So I'm a big fan of experiments, but long-term monitoring is equally important for restoration projects. And as Monique mentioned, because we're part of the National Estuary and Research Reserve System, we're really well poised to put Hester in the context of before and after at degraded control and at reference sites, and really make sense of patterns there. And that includes for our marsh vegetation transects, surface elevation tables that Charlie will be talking about, water quality monitoring, oyster recruitment, shorebird monitoring, and marine mammal monitoring that we do. And just to give you like one example of putting Hester in context, we have vegetation transects at 8 sites around the estuary that are our reference sites to compare to the transects at Hester. And here you're looking at pickleweed height on the y-axis as a function of elevation, and you can see the existing reference marshes show a very different pattern than Hester is so far. In addition to comparing among marshes, you can look within the restoration site and get meaningful information from the spatial variation within the site. So the figure here is from Alex Thompson using our transect data just 18 months after phase 1 was completed. And she found some really interesting patterns. This here on the y-axis is percent cover of the colonizing marsh vegetation. That's pickleweed almost entirely. And she found it was much higher in areas that were former grassland, those scraped areas that were lowered to wetland elevation, than it was in former mudflat or vegetated marsh. That was surprising. Maybe because the sediment addition and compaction affected these marsh areas much more. She also found that marsh cover was much higher immediately adjacent to the creek, but this effect declines really rapidly with distance from the creek. So, you know, that spatial variation really helps provide some clues as to what's going on. Here again, you can see the vegetation hugging the creek edges, and Monique showed you a photo like this too. And a lesson from this for us is that there are climate adaptation trade-offs to different marsh elevations. We built this marsh to be high, most of the marsh plain to be high, as a climate adaptation strategy for sea level rise, but it's actually vulnerable then to another climate-related stressor, namely drought, while the converse is true for the low marsh, more resilient to drought, but more susceptible to sea level rise. Speaking of drought, one of the lessons we learned was that rainfall really matters for high marsh restoration. The best wild seedling germination we ever saw was in this really wet spring where we had one atmospheric river after another. That was 2023. And we had densities of pickleweed seedlings that were crazy and spurgularia too. And this is something that Joy Zedler's lab had studied in Southern California, that seeds need, you know, just the right extended moisture and low salinity conditions to really go crazy like this. But we saw that very clearly at our restoration site. So this got us thinking about what shape does the restoration trajectory actually take at a site like Hester? And how is that affected by rainfall? So here on the y-axis is restoration success. Let's say that's colonization rate by wild seedlings or some indicator like that. Like, I think by default, I just sort of assumed it would be this linear thing that you gradually get more and more over time. But then there's the influence of drought. So that's going to make it more zigzaggy with drought and rainfall years. But also, you know, there's no reason that it needs to be linear. It could be that you get low colonization initially because there's some inhibiting factors, and then due to facilitation or some stressor being ameliorated over time, you get better success later, or you could get exactly the reverse happening. So I encourage you all to think about what is the shape of your restoration trajectory if you're doing a restoration project. So to answer that for Hester, I'll show you just a few examples. We transplanted Disticula saltgrass into exactly the same 60 plots in phase 2 in 3 consecutive years. And we actually found their survival was lower in each consecutive year. So 2022 was right after it was open to the tides, and then 2023 and 2024. Survival of these plants decreased over time. So that makes it look like we may have this nonlinear model going on at Hester, where restoration success is higher initially and then tapers off. Let's look at that with a different data set, actual wild colonization from our transects. And yes, that shape is what we've got. So here we're looking at percent cover of pickleweed from wild seedlings. In green is phase 1. And you can see a nice steep linear increase for a few years, but then it flattens out. And then phase 2, also that nice steep linear increase and slowing down. What we think is going on in that really rainy year I mentioned, 2023, is right here. So we got a greater increase initially and a milder tapering in phase 2 because of the beneficial climate conditions. And Sean will be talking a bit more about this from UAV data. So we developed this into sort of a conceptual model to be useful to others, hopefully, that you could think about, like here, if you have your restoration indicator, like percent cover from wild colonization, in a system like ours, how much cover you end up with may depend just on what your initial rainfall conditions were. If you have your early years be drought, you may reach a lower abundance than if your early years are wet. So Monique mentioned we conducted this restoration in 3 phases. That was primarily for funding reasons, that we had staggered construction in 3 different periods. But actually, with the benefit of hindsight, this is a lesson learned that we would recommend to others, that phasing across different periods yields a temporal portfolio benefit in the face of climate variability. If we build it all at the time we did phase 1, then we would have had all of it facing those drought years initially, while instead, with a mosaic, some of it had this amazing rainfall. All right, so that's multiple phases. Now I want to transition to talking about multiple marsh species, which we've planted throughout, such as here at phase 3 as indicators of those soil addition treatments. We really found that restoring a diverse marsh community is achievable with greenhouse-grown native plants. The plants did not colonize at a high rate on their own, washed in from tides. And so bringing them in with planting was essential for us. I mentioned Karen Tanner and the clustering experiment, and now 7 years later, you still have these nice big patches of Frankenia, of Jalmea, and so on that remain as a result of that initial planting effort. It's patchy, some areas better than others, but only in these planted areas do we have high marsh diversity at Hester. And Jenna Shikuzawa looked at what functions are provided by those plants. And she looked at actually 30 different indicators of marsh function, and I've just shown you 2 as an example here. Above ground carbon content, which Frankenia was best at, and arthropod species richness, which Jalmea was best at. But the takeaway is that no single species optimizes all functions. And so you have multifunctionality increased by having multiple species. So we don't only want diversity of marsh species, we want all the foundation species in this landscape restored. So I just wanted to reiterate, as Monique mentioned, that for us an important takeaway is that you really can take a landscape approach and restore multiple coastal foundation species together. So for us, that was from the coastal prairie grasslands adjacent to the marsh itself, to eelgrass that was planted in Hester Creek that Kat Beheshti will be telling you about in a little bit. And also to native oysters that we established in these new creeks. So we used conservation aquaculture to settle Olympia oysters on native clam shells. These little specks here on the clams are juvenile Olympia oysters. And then this is what they looked like 1 year later when they've grown into these, sort of natural looking clusters of Olympia oysters. And we've actually found on our recruitment tiles that there's really good recruitment in Hester Creek. And since there's no other nearby adult population, we suspect this is sort of a closed system with larval retention that is self-sustaining. So that's for the species we can take credit for. What about the mobile animals? There, we were really pleased to see that if you build it, they'll come, seems to apply. This is a bit of a puzzle. If I had more time, I'd pause longer and let you guess whose footprints we're looking at here. A few people on the call will know. These are sea otter footprints, and we did find that sea otters started using the area, not in high numbers, but occasionally, almost immediately after it was built. Saw one napping on the marsh just the other day, and they're feeding on crabs in the channel. We also see lots of terrestrial mammals, raccoons, of course, here with pickleweed seeds in their footprints. And even in the early stages, right after the marsh was open, there were just pathways of animals all across it, deer, coyote, skunk, and so on. We found that migratory shorebirds really like to roost at Hester when tides submerge the rest of the slough marshes. So here, this is one of the highest tides of the year, and the little peeps still can stand in it because there's just an inch of water, while in the surrounding marshes, there's a foot of water that they could not be standing in. And geese certainly love our marsh, and we have some nesting birds too. Just this summer, we did a nekton study with a graduate student, Carter Newsom, and Mike Polito's lab, and I'm showing you some seining data from seining in Hester versus adjacent marshes. Yellow dots are the fish and crab community from seines in Hester. Green triangles are the fish and crab communities in Yampa Marsh adjacent, a reference marsh. And points that are closer together in this NMDS ordination have similar communities and further apart have different communities. You can see that there's a ton of overlap already between Hester and the adjacent reference marsh. These darker green triangles are a second reference marsh up near the head of the estuary, and Hester is also fairly similar to that. In contrast, our degraded site, Pelican Marsh, has a really different community. So this was actually a bit surprising to me that the fish and crab communities are already so similar at Hester to the reference sites. All right, so we're talking about animals. How about the humans? The human animals, those are my last take-home messages. We've really learned that local communities and tribes are vital partners in the stewardship of these restoration sites. For all of our plantings of thousands and thousands of greenhouse grown plants and monitoring, walking around there with an iPad, collecting data on survival and growth and so on. It's been a huge blessing to us to have all those extra hands and eyes out there. And I think it's been really fulfilling for the community also to feel engaged and ownership in this restoration site. So we're so grateful. I know there's people on the call today who've been out there getting muddy with us, and we're so grateful to the community support. We've worked with various tribes who are connecting to Elkhorn Slough ancestral homelands. In particular, on the oyster restoration, we've partnered closely with the Amamutsan tribal band. They're very interested in returning to a legacy of coastal stewardship of important food species as a part of food sovereignty, and have helped us from the early Hester restoration days get that population established in Hester Creek. And then some of these native stewards are in turn serving as ambassadors to the community and giving presentations to other tribal members about their work and helping in big monitoring events. Restoration sites are also a laboratory for students and other researchers to study coastal dynamics. So this is coming full circle to the start of my talk where I acknowledged Joy Zedler's influence. This was very much the model she used too, of engaging a lot of people and helping do the science at the restoration site. We have 8 publications to date on this marsh restoration area, that the first phase of which is only 7 years old. And that's largely because of all the students engaged their energy at Hester. So now, since Monique and I have covered a lot, I want to just recap our lessons learned for you one more time. As Monique mentioned, robust science was critical for motivating the need for and the design of this marsh restoration project. A collaborative decision-making process supported by working groups, agencies, and communities was critical to success. You can build a high sea level resilient marsh with migration space by recontouring the adjacent hillsides, and wetland species rapidly colonize these former uplands. Scraping sediments from former fallow farm fields provided us opportunities for grassland restoration with a blank slate without an invasive seed bank. And large experiments can be built into every stage, from the earth moving stage to the planting stage to later adaptive management. The results of those experiments may surprise you, which is exactly why you need them. Extensive spatial and temporal monitoring is key to understanding restoration success. Spatial variation within the restoration site provides valuable clues to mechanisms. And there are these climate adaptation trade-offs to different marsh elevations. A high marsh plain is resilient to sea level rise, but vulnerable to drought. Phasing construction across years yields a portfolio benefit in the face of climate variability. Your restoration trajectory may not be linear, so think about what shape it is, and in systems like Hester, get your plants established early. Restoring diverse marsh communities is achievable with planting and yields multifunctionality. And you can take a landscape approach and restore multiple coastal foundation species all at once. If you build it, the animals will come. And local communities and tribes are vital partners in the stewardship of restoration sites. Finally, restoration sites are a laboratory for students and other researchers to study coastal dynamics. And I will end on this slide because it sets the stage for the rest of the day, where you will be hearing from many different students and researchers. So I think I have time for like 1 question or 2 questions.
I've seen only 2 questions so far. The first one was, did you try other irrigation methods, for example, drip irrigation? And
That's
I said,
yeah, you want to answer?
I answered it on that we did the small system, but go ahead.
Yeah, I do think testing different water delivery methods is the key. Like, clearly the plants would benefit from more water available to the roots, but the trick is figuring out how to make that water most available to them, I think.
Yeah, and then the last question was asking for sharing the publication list in the chat or a link to it. So I think we'll have to put that together afterwards.
Yes, I've got it handy. So when we share, I'll send it out with an email.
Fantastic.
Great tip. All right. Excuse me for a moment.